Welcome to our lecture today on drug targets, receptors. So receptors are, they function a little bit as a letterbox for the cell. They are located in the cell membrane and they receive messages from chemical messengers coming from other cells. Those chemical messages are then transmitted into into the cell to affect to give a particular result. Now there's different receptors for specific chemical messengers, and each cell has a range of receptors in the cell membrane that makes it responsive to different chemical messengers. So in this example here, you have a nerve cell. A signal is going to come from the central nervous system. Signal is going to come from the central nervous system, okay, through the nerve cells, through the neurons. And that signal is going to be converted into a chemical messenger. Chemical messenger is going to be released in between the neuron and the cell that's going to be affected. There is a gap. And so that impulse, that information, that signal is converted to a chemical messenger that's then going to interact with a receptor on the surface of that cell. All right. And so once the chemical messenger reaches the cell, it's going to cause a series of other reactions to take place, and that signal is going to be amplified and lead to a cellular response. Okay. So there are many different chemical messengers. Neurotransmitters are some of the chemical messengers, and you also have hormones that are chemical messengers. So beginning with neurotransmitters, neurotransmitters are chemicals that are released from the end of a neuron, and they travel through across a synapse. A synapse is that gap, that space between the neuron and the cell. Okay, so the neurotransmitter is going to travel across the synapse and bind with a receptor on a target cell. The target cell can be a muscle, it could be another neuron, and oftentimes those neurotransmitters are short-lived and they are responsible for messages between cells. So you can think about some neurotransmitters that you know, such as acetylcholine, dopamine, norepinephrine, and epinephrine. And then the second chemical messengers that we are mentioning here today are the hormones. Now, hormones are going to be released from a cell or a gland, and they will travel some distance to bind to a receptor. If the cell that releases, we call them autocrine glands. If the cell that releases the hormone or the gland that releases the hormone has the same hormone working on it, okay. And then you have endocrine glands, where they will release a hormone to go affect a different target. So these are the two types of chemical messengers that we are covering here in this class. Okay. Now, chemical messengers switch on receptors without undergoing a reaction. I also want to mention here that they can also switch off. They can also switch off a receptor. Now, the difference between receptors and enzymes is when a substrate binds to the enzyme, the substrate is actually going to be converted to a product. When it comes to receptors, the messengers are going to bind to the receptors, and they will not undergo a reaction, but they will cause a series of other reactions to take place. That's a major difference between the two. Now, as we mentioned earlier, each cell has a range of receptors in the cell membrane, and they can have some of these receptors are there for neurotransmitters. Other receptors are there for hormones, and so the blood supply is going to provide those hormones there. This is the synaptic gap here. Synaptic gap. And these are neurons. Okay, and these are the neurotransmitters. So each cell has different receptors on the cell membrane, and there are many different types of receptors that we will cover. So the way it works is receptors contain binding sites, as we have covered in earlier lectures. Those binding sites have binding regions, okay? They have binding regions, and they're going to interact with chemical messengers. So there are going to be some intermolecular interactions taking place. Okay. Now the binding of the messenger is going to cause an induced fit in the receptor protein. So once the messenger binds to the receptor, it's actually going to cause some conformational change inside of that receptor, then the change in the shape is going to lead to the receptor being able to bind to other molecules and to cause an effect inside of the cell. Now, the series of effect that takes place, the series of changes that take place in order for the messenger that binds to the receptor to actually cause an effect is called signal transduction. Signal transduction. Okay. And it leads to a chemical signal being received. Now, the messenger themselves don't enter the cell, mainly because of the structure of those messengers and because of the fact that a lot of them are hydrophilic type of molecules that will not be able to cross the cell membrane. So they have to interact with the cell through a receptor, and that receptor then is going to send their message inside of the cell. And that's why we mentioned that the receptors serve as almost like a mailbox for the cell. Okay. All right, so we still continue here with the mechanism of action. As we mentioned, the messenger is going to bind to the receptor, causing a conformational change within that receptor. So it causes an induced fit within that receptor. And once the induced fit takes place, then a cascade of reaction will take place as the signal transduction. Okay, cascade of reaction is going to take place, leading to a message being released into the cell. Now, after the effect is done, the messenger then is going to be removed from the receptor, and these reactions can go on and on and on and on. Now, focusing a little bit more on the binding site of these receptors, oftentimes they are hydrophobic, hollow, or cleft on the receptor surface. They're equivalent to the active site of the enzyme, and it is at that place that you are going to have the binding of the chemical receptors. Now, we are talking about chemical receptors here, so there's going to be chemical interactions. Okay, chemical interactions, and the chemical interaction is going to take place through amino acids. So these are the amino acids that are actually going to bind with the messenger. Okay, again, no reaction or catalysis take place at the binding site. Only the chemical interaction, the hydrogen bonding, the ionic interactions, and hydrophobic types of interactions. As soon as it binds, it causes a conformational change inside of the receptor. Okay, so let's suppose that we have a messenger that's looking at this particular binding site here. When it approaches, when the messenger approaches the binding site, the receptor is going to undergo some intermolecular conformational rearrangement so that it can fit that messenger, and that's what we call the induced fit. Okay, so the binding site itself is not the correct shape. It's nearly the correct shape for the messenger, so it alters its shape. The binding alters the shape of the receptor so that it can fit the messenger, and then that leads to further effect, signal transduction. Okay. Now, we've mentioned these binding interactions multiple times: ionic, hydrogen bonding, van der Waals. So if this is the binding site, for instance, and you have inside of that binding site some specific binding regions and some specific amino acids that are going to interact. So based on the types of amino acids that you have, you can already predict the type of reaction that are going to take place. With the more hydrophobic with the more hydrophobic moieties, you have van der Waals interaction. With the ionic moieties, you can have ionic bonds, and hydrogen bonding can take place where it's possible. Again, the concept of induced fit, it happens because, as we mentioned before, the binding site itself at first is nearly perfect, and perfect is probably not the right word, but is nearly has the shape that's necessary to accommodate the messenger. As the messenger comes closer and these interactions start taking place, the binding pocket is going to rearrange its shape so that it can fit the messenger more tightly. So the induced fit is a way for the cell to maximize the intermolecular bonds and optimize the interaction between the messenger and the receptor. So let's talk about the overall process of receptor-messenger interaction. So you have a receptor, you have a messenger there. Induced fit caused by the binding of the messenger, which is going to lead to signal transduction. At the end of that signal transduction, there's going to be a release of the messenger. Now, there is a balance that needs to take place here. Similarly to what we've seen with enzyme, the binding interaction has to be strong enough to hold the messenger sufficiently long so that you can have a reaction, but it can't be too strong, right? It has to be weak enough to allow the messenger to leave as well. So there's a balance there that needs to happen. Now, when you look at a particular receptor that you want to block, if you want to design an antagonist for a receptor, you have to take into consideration the types of binding interactions that are involved at the binding site. There are three big classes of receptor superfamilies that we'll look at in this class. There's the ion channel receptors, the G protein coupled receptors, and the kinase linked receptors. And there are also some intracellular receptors that we will cover. Between the G protein coupled receptors and the kinase receptors, you have two of the most important targets for drug design and action. The ion channel receptors are also very important. Now, the amount of time that it takes to the response time is different. The ion channel receptors response time is within milliseconds. G protein coupled receptors within seconds, and kinase link receptors are within minutes. Intracellular receptors have longer response times. So if you think about an intracellular receptor, the steroid receptors, the thyroid receptors, those can have effect that last longer, last days. So ion channel, G protein coupled, and kinase link receptors are membrane bound. All right, so let's begin with the ion channel receptors. So the receptor protein is going to be part of a ion channel protein complex. Now, why do we need ion channels? Because ions are charged. They're charged. They're charged. They have a positive charge or a negative charge on them, and they will not be able to cross that cell membrane. So you need channels that will allow those ions to cross. There are two types of ion channels in general terms. You have the ligand gated channels, and then you have the voltage gated channels, which function based on the gradients. Okay, so again, the receptor is going to bind to a messenger, leading to an induced fit, which is going to cause ion channels to be open. Now those are very specific type of channels, okay, and very specific type of channels. And these are important, especially when um, you're dealing with heart rhythm, or you're dealing with diuresis, trying to reduce blood volume. Um, it's important to be able to block specific ion channels and to prevent water reabsorption and things of that nature. So normally ions will flow across the cell membrane. They'll flow across the cell membrane down a concentration uh, gradient, and you will have to polarize or depolarize nerve membranes. As we've seen before, when you activate a receptor, you can either activate or deactivate an enzyme catalyzed reaction within that cell. Now, so looking at this, looking at this here, so the ion channels are actually going to allow charged charged ions to be able to go through a hydrophilic tunnel and pass through the cell membrane, which is hydrophobic, okay, which is hydrophobic. So those ion channels really provide, if my pen wants to work, it's very, very slow right now, provide safe passage for these ions that would not otherwise be able to go through the cell membrane. Now, looking at the tunnel this way is a bit misleading, right? It looks as if the tunnel is open all the time. The reality is there is a gate. It's a gated type of channel. Okay, it's a gated type of channel. And the ion channel is usually closed unless you have a chemical messenger that binds to the receptor and then causes the gate to be open. Okay. So the chemical messenger comes closer, it binds to the receptor and causes an induced fit, which leads to the opening of the channel. Now let's look at the nicotinic receptor. The nicotinic receptor is set up in such a way that you have 2 alpha subunits, 1 beta, 1 gamma, and 1 delta. Okay. The binding sites, as you can see, are located right there in the red. And the ion channel. So if you were to flip this, and have the top of the subunit face you, you'll be able to see down the ion channel. So this is an ion channel that is activated by the this is the ion channel that's a nicotinic receptor. Now, some of these receptors, such as nicotinic, muscarinic, were given names based on natural substances that will have an effect on them. Okay. So there's 2 ligand binding sites that are mainly on the alpha subunits. Okay, that's for the nicotinic receptor. Then you can also have a glycine receptor. Glycine receptors are different. They have 3 alpha subunits and 2 alpha subunits, and the binding sites are located on the alpha subunits. And you also have the 4 transmembrane receptor units. Okay, and the way they're organized is the neurotransmitter is going to have a binding region, okay, usually in the N terminal. Then you have these transmembrane proteins here. You have an extracellular loop, intracellular loop, and then you have a variable loop. The variable loop is so called because the length varies. So the length number of amino acids. Okay, it varies depending on the specific channel that we're dealing with. Now the 4 transmembrane regions are hydrophobic. So let's keep looking at the detailed structure of the ion channels. When you look at the ion channels, you have protein subunits. Okay, so you have these protein subunits, and then you have the transmembrane regions. So 1, 2, 3, 4. These are the transmembrane regions. And just remember region 2 of each protein subunit lines the central pore. Okay, so you have, here is TM2, just remember 2, 2, 2. They all line the central pore, creating that tunnel for the ions to go through. So looking at the 5 glycoprotein subunits here traversing the cell, you have a messenger that comes, it binds the binding site of that receptor, which leads to an induced fit, causing the channel to open up, allowing passage of ions. Okay. So we mentioned earlier nicotinic receptors, for instance. They are cationic channels for potassium, sodium, and calcium. Then you also have anionic receptors, which are inhibited by GABA aminobutyric acid. Okay, gamma aminobutyric acid, GABA. So the binding of the neurotransmitter or the binding of the chemical messenger can either be inhibitory or it could be excitatory. Now you look at the ion channels here. So here we're only looking at the transmembrane protein 2. The transverse view shows you how these proteins are arranged to when it's closed, and then it shows you how they, after the binding of the messenger, those proteins actually rearrange themselves, the induced fit, to allow for the ion flow. Okay. Again, the ion channels have a really fast response. They're really important for transmission between nerves. Okay. And binding leads to ion flow across the cell. Now, the flow of ion is what we call the secondary effect, and signal transduction. Obviously, this is going to change the concentration of a specific ion within the cell and leads to variation in cell chemistry, but it's also going to lead to variation in physiology, right? Physiology. Let's suppose that you block sodium reabsorption, sodium reuptake in the kidney. If you block sodium reuptake in the kidney, that means you are also blocking water, because water follows sodium. You're blocking water. If you're blocking sodium, you're blocking water. You're reducing blood volume. Okay, you're reducing blood volume, you're reducing blood pressure. Okay, so not only does it change the variation in cell chemistry, it also changes the physiology of the whole body. Okay, now that we've looked at the ion channels, let's take a look at the G protein coupled receptors. Again, it's the same idea where the receptor binds to its messenger, leads to an induced fit. The G protein coupled receptors are called so because the receptors have the ability to bind. The receptors have a binding site, an intracellular binding site that binds to a complex that has a binding site for GTP and GDP, and you will look into it in more detail in the coming lectures here. So once the messenger binds, then G protein is going to bind as well to that receptor, and then it's going to be destabilized, and you are going to have a disintegration of the G protein complex. You have 3 different subunits that are going to be disintegrated, leading to signal transduction. Okay, so the G protein is a 3 subunit protein. Okay, and it's the alpha subunit that binds to to GDP and GTP. All right, so this is the G protein coupled receptor. Okay, it binds to a messenger. When it binds to the messenger, it's going to cause an induced fit, causing the binding site here to be open, which will then lead to this G protein complex here to come in and bind in the binding site. And once binding takes place, the alpha subunit is going to disintegrate itself and continue the signal transduction. Okay, so the alpha subunit is going to disintegrate and continue signal transduction. All right, so as we see here, the G proteins don't bind themselves to where the messenger binds. They bind to an allosteric site. Okay. The induced fit is going to result in the opening of an active site, and then an intracellular reaction is going to be catalyzed. Again, so active site is closed. So let me recap quickly. So you have a chemical messenger that binds to the G protein coupled receptor, which causes an opening of a site where the G protein complex is going to come. The G protein complex comes and binds to this complex. Then there is a rearrangement within the G protein that leads to a weakening of the complex, and then the alpha subunit of the G protein, is then going to be released to go bind to an allosteric site. Okay. Once it binds to the allosteric site, it opens up the active site of the enzyme and leads to intracellular reactions. So the G protein coupled receptors have seven transmembrane regions. Okay. And you have this N-terminal, the C-terminal, and the extracellular loops and the intracellular loops. Again, you have a variable intracellular loop here. As we mentioned, the variable intracellular loop is there, so-called because the number of amino acids can change, the length of the loop can be different. Then there's a G protein binding region here. And depending on the type of G protein coupled receptor, the chemical messenger will bind at different places. So those chemical messengers that affect the G protein coupled receptors are monoamines, dopamine, histamine, noradrenaline, norepinephrine, and acetylcholine. All these are examples. Then you have nucleotides, ATP, GTP, GDP. Okay. You have lipids, you have hormones, glutamate, calcium. All these are potential ligands for the G protein coupled receptors. Now, as I was mentioning before, the binding site depends on the receptor type. So the monoamines are going to bind in a pocket, within a pocket in the transmembrane helix. Okay. When it comes to the peptide hormones, they're going to be more on top of the extracellular loops in the N-terminal. Let me label this N-terminal. This is the C-terminal. Okay. Hormones are going to mainly bind in the extracellular loop on top of the at the N-terminal chain, and then glutamate is going to mainly bind on the N-terminal chain. So different ligands have different binding position. Okay. Now, rhodopsin is a type of G protein coupled receptor, so-called because rhodopsin is a visual receptor. It's involved in the ability to see There are many common receptors that belong to this family. And when you look at the, I don't believe I included it in this lecture, but when you look at the evolutionary tree of the G protein coupled receptor, you can look at them and realize that some of them are really close in structure. So, for instance, you can have H2 acetylcholine, dopamine, very close in structure because they're very close in their evolutionary within the evolutionary branch. So when you are designing agonist or antagonist for one type of G protein coupled receptors, we have to be careful to make sure that it is specific enough to that for that receptor so that you don't elicit side effects. Okay. So those G protein coupled receptors were actually difficult to crystallize. However, bacterial rhodopsin is basically rhodopsin from a bacteria, was used as a template for those receptors and was used to solve the structure of those receptors. So a lot of models were based on the template of amino acids from the bacterial rhodopsin. This allows us to have a certain understanding of the binding site so that we can design drugs to either inhibit or activate those binding sites. Okay. I actually included it. So this is the evolutionary tree of these receptors. Just something I want to mention here. You look at these muscarinic receptors. We mentioned that some of the receptors are so-called based on the types of natural substances that activate or inhibit them. So you realize that for the muscarinic receptors, for instance, muscarinic 1, M1 receptor, and H1 are actually closer when it comes to the evolutionary tree. So it is possible to develop, for instance, a molecule that's going to bind to the H1 receptor that is also going to be able to affect the M1 receptor and the H2 receptor. And similarly, it's possible to have the H2 antagonist or agonist that's also going to affect the alpha 1 receptors. So it is important to keep that in mind as we strive to develop drugs that interact with these receptors. Okay. So this classification here basically is a reflection of which type of receptors recognize the same ligand. Okay. So adrenergic receptors, alpha and beta. Cholinergic receptors, you have nicotinic and muscarinic. And then you have all these different subtypes there. Okay. As we've mentioned, for the muscarinic, you also have the H1 receptors that are muscarinic, right? And then you have the histamines, histamine receptors. This is important to know that those receptor types are not equally well distributed in tissues. So some places in the body have predominantly a certain type of receptors. Okay, so for instance, in the GI tract, you have the beta 2 receptors are present in both the GI tract and the bronchial muscle. Okay, and in the heart muscle, you have mainly beta 1 adrenergic receptors. So in order for us to develop a particular drug that's going to target, let's say, fat cells, one of the ways to do this is to target selectivity to beta 3 adrenergic receptors. Now, kinase linked receptors. Kinase receptors are really interesting because they're activated by They're activated by hormones, and their overexpression can result in cancer. So some of the anti-cancers that are on the market actually target tyrosine kinase linked receptors. Okay, so the protein serves the dual roles of a receptor and an enzyme at the same time. The receptor is going to bind to its messenger, leading to the induced fit. Protein changes shape again, opens the intracellular active site, and then the reaction is going to be catalyzed within the cells. Similar to what we've seen before, so the messenger binds, causing opening of the site inside of the cell intracellularly, which is going to lead to intracellular reactions. And we will cover those intracellular reactions in the next lecture. Once the messenger leaves, the site is closed. Now, the tyrosine kinase receptors have an extracellular N-terminal chain, and the intracellular C-terminal. They have a ligand binding region and then a catalytic binding region. So this region is closed when it's in the resting state. It opens up once you have ligand binding. Then you have the transmembrane region, which is hydrophilic transmembrane region. So these tyrosine kinase linked receptors are going to phosphorylate, cause the phosphorylation of tyrosine. Okay, cause the phosphorylation of tyrosine, and you can also have other groups such as threonine that can also be phosphorylated by kinases. Okay, so the epidermal growth factor receptor is a very specific type of tyrosine kinase linked receptor. So the epidermal growth factor is going to bind to the binding site here, causing a dimerization. As you can see, when it comes in, it's going to cause those two units here to come together for a dimerization. These groups here, the hydroxyls, are actually going to be phosphorylated. Okay, so it's going to cause phosphorylation. So epidermal growth factor comes in, binds at the binding site, causing dimerization. Then the induced fit is going to open the tyrosine kinase active site, leading to phosphorylation of the tyrosine. So on this particular, for this particular mechanism, the active site on half of the diameter of one is going to catalyze phosphorylation of the tyrosine residue of the other, and vice versa. Okay. As you can see, the dimerization is very crucial, and the phosphorylated regions are where other proteins and other enzymes are going to bind to lead to more signal activation and signal transduction. Okay. This will lead to the message being carried into the cell. The insulin receptor is another type of tyrosine kinase linked type of receptor. Again, you have a binding of insulin to its receptor that's going to cause dimerization, and the kinase active site is going to be open by induced fit, causing the phosphorylation of the hydroxyl groups. Okay, and again, the catalytic site of one subunit is going to phosphorylate the tyrosine on the other residue. Growth hormone receptor is another type of tyrosine kinase linked receptor. So in the presence of growth hormone, you have a a dimerization that takes place, and then the kinases are going to bind to the receptor. The kinase active site is going to be open, leading to phosphorylation. Okay, so growth hormone receptor is another example of tyrosine kinase linked receptors. Now, to complete our conversation on receptors, we will include the intracellular receptors. So in this particular case, the chemical messengers have to cross the cell membrane. They have to cross the cell membrane. Those chemical messengers have to be hydrophobic, and so that they can cross the cell membrane. So examples are steroid hormones, and so the structure the structure of those hormones have oftentimes you have a zinc that's present there. You have a DNA binding region that has a zinc finger. Then you have a steroid binding region. And then the zinc finger contains cysteine residues, and the presence of the zinc allows, facilitates the conformational organization of the particular protein. Is going to allow for interaction between the sulfur and the zinc. So let's talk a little bit about the mechanism. So the messenger is going to cross the cell membrane because in this case it is actually hydrophobic. It's then going to create a receptor ligand complex, leading to a dimerization of a receptor ligand complex. A coactivator protein is going to be added to that complex, which is going to lead, which is going to bind to DNA, then leading to transcription being switched on or off. Okay. Now, with transcription being switched on and off, it will lead to either protein synthesis being activated or inhibited. The estrogen receptor is one such receptor. So you have the binding site. Estradiol can come in and bind to that binding site, which is going to lead to dimerization and then exposure of the AF2 regions. Okay, the AF2 region is where the coactivator is going to bind, and you're going to have recruitment of the DNA, which will then lead to transcription. Okay. Okay, so this is a quick run through chemical messengers and receptors. The next lecture we are actually going to cover signal transduction. Thank you.